our class now. It is already 7:47. Meanwhile, whoever wants to join, they can. So, in today's class, we will start another topic basically. In the previous classes, we saw how to prepare a scaffold for your tissue engineering construct, right? And there are various scaffold fabrication techniques. Then, what kind of materials you can use to prepare those scaffold, and then when you are trying to mimic the in vivo environment, you are basically trying to grow the cells in an environment that is like found in your body. So and how will you actually say that the cellular behavior is exactly like your in vivo environment, right? So for that we have to do some studies, characterization studies and all, to assess how our scaffold is modulating the cellular behavior, and is it same as that found in the body, right? And then we saw basics in the initial classes, we saw basics of cell biology, etc. Now, if you remember our initial discussions on like tissue engineering, like introduction to tissue engineering, if you remember that, there we discussed that the tissue engineers, they want to mimic the in vivo environment or the in vivo tissue environment or organ environment. And we know any tissue is composed of cells and ECM, and the cells, they communicate through signaling molecules, right? So what we can do, we can, yes, scaffold can we can prepare, that is the artificial ECM we can prepare. Then we can take cells from directly from the body, right? Like the primary cells, or we can use stem cells, etc. But in order to direct the cellular behavior into the direction that we want, okay, into a desired direction, we have to take help of signaling molecules, right? So that is what we will discuss, how we will deliver these signaling molecules into a tissue engineering construct, so that these molecules can guide the cellular behavior, right? So the tissue engineering triad means any tissue engineering scaffold is basically has three components that we already know: cells, scaffold, and signals. And the activity of the cells, they are controlled by signals. And signaling is important because we know cells can communicate with each other. Like there is direct cell-cell communication, or cells can secrete some molecules, signaling molecules, and the neighborhood cells will be alerted through these signals, or the environment, like through ECM as well, cellular communication is possible. And we can we have to mimic either of these cellular communication types in our tissue engineering construct to get the desired function out of your tissue, okay? So there are different types of signals available to us. So there are the chemical signals, which we know, the growth factors, then the mitogens, morphogens. So mitogens are basically molecules that initiate cell division, and morphogens are those molecules that are released during tissue development. Okay. And there are other types of signals as well, like mechanical, that we already see that how cells can respond to mechanical signals. That is called a new field, mechanotransaction. And cells can also respond to electrical signals, which we didn't see previously, but it is possible. But in today's class, we will focus on delivering chemical signals only, that is delivery of growth factors. Okay, into our tissue engineering construct. So what are growth factors? Let's recall. So growth factors are small protein molecules which are soluble in nature, and these growth factor molecules are produced by cells, all right, and they can act as a signaling molecule between two cells. That is, we can initiate paracrine signaling, right, or autocrine signaling. So these kind of signaling modes are possible using growth factors. They are like known to help in proliferation, cellular growth, and differentiation. Now, I have given a small note for you because recently I found this information and I thought it is worth mentioning that the term growth factor, we often use it interchangeably with the term cytokine. So maybe in my previous discussions, I may have used it as well, but they are not same all the time. Okay, it is not like uh, growth factor and cytokine. They cannot be same at all circumstances. So growth factors are like given the name growth factor is given to those molecules which are known to have an positive impact on cellular growth. Means they only promote cellular growth. Whereas cytokines, uh, they are known different there is a various family cytokines are there, right? So under this broad umbrella of signaling molecule that is cytokine, people have noted that these molecules have positive, negative, or neutral effect on cellular growth. So growth factors are those molecules which positively affect cellular growth, whereas cytokine doesn't necessarily have to be give us positive effect, rather it can give negative effect or neutral effect. So you can say that all growth factors are cytokines, okay, but all cytokines are not growth factors. So in tissue engineering, we will be using this term growth factor to refer to all the signaling protein molecules, which is like affecting cellular growth, migration, proliferation, and differentiation. So this confusion that is there between the usage of these two terms is because the term growth factor is coming from like the field of developmental biology. Okay, and the cytokine, the term cytokine is coming from the immunology. So when people studied um, a few molecules which are cytokine as well, okay, and they were seen to show positive effect on growth, so they were like interchanged, like that is why this confusion happened. But in tissue engineering, we will only use the term growth factors to refer to all the proteins which is affecting these type of cellular fate processes. All right. Now, why should we use growth factors? So maybe it is obvious to you by now, given how much we have learned so far, but still, I will discuss it. So in tissue engineering, we understand that there is an obvious advantage to using biological materials like a cell for treating a disease. Correct. So it is advantageous to deliver cells, healthy cells, at a disease site. Now, having cells in the scaffold would mean that the cells are able to produce their own growth factors and they will initiate proliferation, means through these growth factors, they will initiate proliferation of the host cells. Thereby, it will improve the healing time. So, cells are themselves the source of growth factors, right? So, why would we then need to deliver additional growth factors? But tissue engineers also work with those constructs which are cell-free, means only scaffold is used for a particular application, no cells are there. So for these kind of constructs, growth factor is important because without the signal, the source of signal, growth factors are what? If you supply growth factor, that becomes the signal, and then it will like diffuse to the nearby, say, host area, host tissue area, and will signal the host cells to come and adhere on the scaffold and improve the healing time. Okay, so it is basically use of growth factors is like essential, or it can you can say it is helpful when you are using only scaffold for a particular tissue engineering purpose. Now, if you are now another question becomes obvious, like people are also using growth factors along with cells in the scaffold, like they're incorporating all the three triads of tissue engineering. Why? So using of growth factors, it can basically enhance your, say you are making a tissue engineering scaffold and you are making it for the purpose of regeneration of tissue. So using growth factors, it will basically enhance that regeneration process. You can show better results, okay? Because cells are means cells, say you want to you're using some cell type and you want um, those cells to say show angiogenesis process. Okay, that is you're using endothelial cells, so you want angiogenesis process to happen. And you know VEGF, vascular endothelial growth factor, so that is known for the process of angiogenesis, means it can promote angiogenesis. So usually at the wound site, this VEGF concentration is already there. Okay, that is how the broken of capillaries, blood capillaries, they will start forming new blood capillaries. Now, if your target is to accelerate the healing process, so your concern should be also to building up the blood vessels as fast as possible, so that the tissue that is in process of like regeneration, it can receive nutrients and oxygen as fast as possible. Okay, so what you will do, you can then engineer your tissue scaffold where you are using endothelial cells, but you also deliver VEGF. Okay, so what that it will do, it will not only allow angiogenesis process for the endothelial cells to occur, but will also allow the host endothelial cells to like start the angiogenesis process earlier, and then the blood vessel formation process, that is angiogenesis process, it will happen at an accelerated rate. So in this way, we can enhance our like regeneration process. Then we can also use these growth factors for differentiation purposes. So mesenchymal stem cells. Okay, you have obviously heard. I have discussed this with you. So if you want to say direct it to become osteoblast, that is the bone cells or osteocyst, then growth factors like BMP, etc., they are incorporated within the scaffold. Okay, so they incorporate mesenchymal stem cells and growth factors like BM2, etc. And then they show that from the mesenchymal stem cells, you are able to get bone formation at your injured site. So for these kind of applications, growth factors becomes like advantage for the engineer. Use of growth factors is advantageous. All right. Now, how growth factors act? Now, like all signaling molecules, these growth factors are capable of binding to receptors which are present on the cell surface. And once they do that, it will activate a intracellular signal transduction pathways. So it's relatively easy to understand. So basically, receptor ligand interaction takes place, and a signaling pathway gets activated. But growth factors can't act in an endocrine fashion. Means like the hormones which can circulate in our blood vessels, growth factors can't do that because they are since they're of very small molecular weight, so their half life is very low, means they will degrade very fast, and the diffusion is also happens very slowly. So the effect of the growth factor is limited to a small area only. So what can we understand from this line? What we can understand is, if we are thinking about delivering some kind of growth factor, we have to make sure that if we are the target, if we deliver it at the target location, okay, we don't deliver it too far from the target site. So its effect, if we, its effect will then reduce. If we deliver it further away, its effect will reduce. Also, if we can like protect the growth factor from degradation in some way, so that the half life of the growth factor um, is not basically stability of the growth factor is increased, then it will be much more useful for us. Okay, so from these understanding, we will see how they are delivering growth factors. Also, we have seen, when we are talking about cellular communication and how cellular fate processes are coordinated using growth factors, we have seen that there are multiple ways through which a growth factor and its receptor, it can interact with one another. If you recall one diagram where there were soluble receptors and membrane bound receptors, okay, and how signaling will happen, and in which cases signaling will not happen. Similar thing is mentioned here. So one growth factor, it can activate multiple receptors, right, simultaneously. Okay, and then also each growth factor, it will have different effect on a different cell type. Then ECM, the target cell location, the concentration of growth factors, they will all determine or help us determine how the growth factors will finally act on the like desired application, say wound healing, etc. Okay, so clinically, people are using growth factors. How? So these are some of the examples. After chemotherapy, when the patient has a low WBC count, then the patient is given with the growth factor GCSF, is granulocyte colony stimulating factor. So by injecting, they injected basically into the bloodstream directly, and through this growth factor, doctor is capable of increasing the WBC count in the patient. Similarly, when during bone marrow transplantation, if you remember, where before chemotherapy or radiotherapy, the mesenchymal stem cells from the bone marrow, they are removed. Okay, so this process of removing from the bone marrow is quite painful. So what another alternative approach of this is, the patient is injected with a growth factor GM-CSF, that is a granulocyte macrophage colony stimulating factor, and this growth factor triggers the stem cells that are there in the bone marrow to come out from the bone marrow and be available in the bloodstream, so that a doctor can directly take out the blood containing the stem cells of bone marrow. Then another example is the growth factor erythropoietin is often used to increase the IBC count in anemic condition like maybe severe anemia. Similarly, this growth factor oprelvectin. Okay, so maybe I pronounced it wrong. Maybe you can check it in online. What is the correct pronunciation? So this growth factor is used to increase the platelet count in thrombocytopenia disease. Okay, but there are some challenges related to the applications that I just mentioned. One is we have to be very like fixed about the fact that the growth factors have a very small half life, and its action is limited to a local area. So we have to deliver it to its intended site. Okay, we cannot deliver it very far away. Otherwise, we cannot see any effect at the desired location. So what we can do is, we can then directly inject, right? We can directly inject at the target site. But again, if the target site is say pancreas, right, or your heart, so it becomes and usually people use very high doses of growth factors for because again, because the half life is short, so rapid degradation will happen, and if you want to see some long term effect, you have to use a higher dose. So in those cases, directly injecting into a vital organ, okay, and that too in high dosage, is risky. It can cause side effects in patients like cancer, right? So direct injection is not a good solution for delivering growth factors at a target site. What can be the alternative approach? We can use advanced delivery systems which will encapsulate the growth factors, and they will be functionalized in such a way that through the blood circulation they will reach the target site and will show action only at the target site. If it strays away from to some another organ location, then the encapsulated growth factors won't be released. So these kind of advanced engineered delivery systems can be used for delivering the growth factors. Any queries so far? I hope it's relatively easy till now. Monday, I have
Yeah.
okay. I have received your group's report. All right. Only
Okay, ma'am.
only one more group is left, so they will submit after 27th. Yeah, 27th they will submit. Okay. So growth factor delivery system. So what are the advantages of using these kind of engineered advanced delivery system? First is using these systems we can achieve spatially controlled release of growth factors as well as temporarily controlled. Means we can see or we can trigger the release of growth factors at a particular time point, and we can control where the growth factors have to be released. Then we can achieve sustained release rather than burst release, where instantly there is a spike in growth factor concentration. We can achieve sustained release so that the effect of growth factor at the target site will be for a longer period of time. Then we don't need very high dosage of growth factors for seeing effect. We can work with smaller concentrations as well, because when you talk about what is the source of these growth factors, it is usually like in recombinant proteins. They are usually recombinant proteins, or they can be taken from another patient, and maybe from serum, etc. And they are purified through various processing steps. So the cost of these growth factors, few mgs, is very high. So when we are talking about treating large number of patients at a lower cost, then we cannot really afford to inject at high dosage. We want to use smaller doses of growth factors to reduce the overall cost of the treatment. Then targeted delivery is possible. As I mentioned previously, we can functionalize the delivery system to release the payload, that is a growth factor, at the target site, and thereby the side effects which can happen due to like delivery at the wrong organ site or tissue site, that can be circumvented. Also, like the encapsulated way of delivering growth factor will protect your protein from the proteases which are there in the body and prevent its degradation. Okay, or reduce its rate of degradation. So that is another advantage of using these kind of delivery systems. So what are the approaches generally used? There are three approaches. One by one, we will look into each of them and we will see some examples under each of them. One way is you immobilize the growth factors directly onto your tissue engineered scaffold. So you have prepared a scaffold through any of the techniques that I previously mentioned. You have seeded your cells, and you use some technique to immobilize, means fix your growth factor molecules onto the scaffold. So what will happen? You're trying to basically mimic the ECM structure, right? So in the ECM, uh, it is possible for the growth factors to stay there so that the nearby cells will get a constant signaling. Okay, constant signal in the cells will be on. Okay, so this, and cell ECM interaction will happen, and yes, the constant signal will be keep on going to the cells. Now, another approach is use nanoparticles to deliver these growth factors. And third approach is use genes. Like the gene which is encoding for your growth factor. So rather than delivering the protein, which has a lower half life, you can use the gene encoding that protein, and modify the cells at the gene level, not at the cytoplasm or cell level, at the gene level. So, using these three approaches, we can ensure delivery of a particular growth factor. You can say we can ensure, or we can use these growth factors to signal our cells and change their behavior into the direction of our application. Okay.
Ma'am, the last slide, there is mentioned, right, temporally and spatially. It means introducing the growth factor from different parts of the body, ma'am.
Where? Which point?
The first point, ma'am, temporarily and spatially controls. The spatially
So,
controlled means, ma'am.
spatially means within the space, like in the body, different organ site. That is what meant. So we can actually control at what organ location the growth factor is released. Okay, and also at what time point. We can give some
How we can do
stimulus.
that, ma'am?
So we can use like if we know some protein biomarkers which are there in a tissue. So our growth factors can be functionalized to release the payload when those biomarkers are there. One example I can give is the cancer site, that is a tumor site. The pH of those site is relatively lower than our body's pH, means there is an acidic environment. Okay, so it is the pH is not 7.4. So we can use a material to make say a nanoparticle out of it and load our growth factors into that nanoparticle. And special property of this material can be to change its porosity when appropriate pH is there. So in our case, if the acidic pH is there, then the pores will open, means the pore size will enlarge. Okay, so there will be high release in growth factor. But if the pH is near physiological range, then the pore size will be relatively smaller. So the release of the growth factors will be very less. So in this way, we can use this and nanoparticle to deliver the growth factor at the um, cancer or the tumor site. Okay, so most of the knowledge that we have about delivering the growth factor comes from the drug delivery systems. Okay, so drugs are also like encapsulated within some carrier, right? So people have taken a lot of information from there. Okay, so this is one way to control where the growth factor is released and at what time. When
Okay, ma'am.
okay. So first approach is as I said that we can immobilize or encapsulate our growth factors onto a matrix. So in this image, what I'm trying to say is, suppose uh, you so in this side, the cells, it has a receptor. When growth factor is there, it will respond to it. Okay, so you will see without immobilization when growth factor is not immobilized. So and cells are there into your scaffold. In the scaffold there is the cells, and you're externally providing the growth factors, and to that the cells is responding. But in the scaffold, if you already immobilize it, then you don't have to externally provide the growth factor. Rather, as soon as you see the cells onto a scaffold, some signaling process will trigger inside the cell. So that is the advantage of immobilization over not immobilizing it. Okay. Now there are three approaches through which you can immobilize your growth factor onto your scaffold. One approach is to physically encapsulate it. So there are also various ways how to do that. But basically, what people do is they use a polymer solution and like add the growth factors into it, make a composite kind of a thing, and then the scaffold along with the polymer is then cast into a 3D structure. Okay, that is the scaffold where the growth factor is there in the bulk of it, and then it is transplanted. So and then some effect can be seen at the transplanted site. Another way is chemical conjugation. rather than relying on the physical interactions like hydrogen bonding or electrostatic interaction, etc., between the growth factor and the scaffold. You can chemically link them, like you can create covalent bonds between the growth factors and your polymer scaffold, so that you can have a better control over the its release. And the third approach is take inspiration from the native ECM structure. Okay, means you try to mimic that ECM structure and use the knowledge of how growth factors are distributed on the ECM. In that way only, you distribute your growth factors into your artificial ECM, that is your scaffold. That is the three approaches. So we will see what are these approaches in depth one by one. So the first one, that is physically encapsulating it. Uh, One way of doing it is, as I said, you take your growth factor solution. It usually is in liquid form, and then basically mix it into a polymer solution. Okay, and you have to do it when the polymer is in liquid state. And after it becomes a gel-like structure, means after it becomes a hydrogel, you cannot do that. So there are advantages, like as it sounds, it is easy to do. Okay, as easy as it sounds, it is that easy. Okay, and the properties of the scaffold and the growth factors, they are not changed. You are not hampering the activity of either the polymer material or the activity of the growth factor. But the disadvantage is loading capacity is low. Means there is a limitation up to how much growth factor concentration you can load, and that will give you repeatable results. Okay, so the loading capacity is low in this in this particular type of encapsulation method. Another way is you can absorb your growth factor, sorry, absorb, okay, absorb, not adsorb. So you can basically make your growth factor solution or the growth factors to absorb, okay, onto this matrix. So you make your scaffold, say using freeze drying technique, and then you add drops of growth factor onto this surface. Then what will happen if this your scaffold is hydrophilic, right? Then it will allow easy absorption of the growth factor, and basically wherever you have dropped your solution, it will get absorbed only there. It will not migrate throughout the bulk, rather it will absorb at the localized site. So again, the advantage is it is easy to do that, but disadvantage is we don't have very good control over how the absorbed growth factors will be released. So there's a possibility that when you transplant this growth factor loaded scaffold at the site, there will be burst release. Okay, so burst release means you will see a sharp peak. So this kind, and you will get an instantaneously high concentration of growth factor, and it might be like toxic to that tissue area, okay, and cause cell death rather than, and you wanted to promote cell growth, you have rather started like cell death or mutation, you can generate cell mutation. So that is why we don't prefer for a technique which gives you burst release. We always go for a technique which will give you a sustained release. Then another way of physically encapsulating your growth factor is by layer self assembly. So for doing this particular technique, people use a class of polymers called polyelectrolytes. So polyelectrolytes are generally charged. So you will find polyelectrolytes of different charges, like they can be positively charged, like cations, or negatively charged. And you can layer these polyelectrolytes. Okay, and in between the oppositely charged, so you can first give a layer of, say, positively charged polyelectrolyte, then you like give your growth factor molecules, and basically trap your growth factor molecules in between positively charged polyelectrolyte and negatively charged polyelectrolyte. So the due to strong electrostatic interaction, the two types of polyelectrolyte will not separate easily, but at the same time, it will trap your growth factor molecules in between. So the advantage of using this type of physical encapsulation is you can have a better control over the delivery rate. How you will control it? So you can basically play with how many layers you want in your structure, right? So if the number of layers is high, right, uh, means you can use two layers of positively charged binded with two layers of negatively charged, right? So in this way, your growth factor molecules will be much tightly trapped. But if you use opposite, means you use less number of layers, so then the release of the growth factor, or it is easier to break the electrostatic interaction and get your growth factors out. And people have observed that using this technique, the burst release is minimal. Okay, so burst release is there because it is physically means physical interaction is there, so that is relatively weaker than chemical interaction. So burst release will be there, but it is minimum to a minimum extent. So this is just an image way of showing, like image representation of showing all the three types of physical encapsulation technique. So in this, here within the bulk of the hydrogel, you have encapsulated your growth factor molecules by directly mixing it, or you prepared your hydrogel scaffold or any polymer scaffold, and on top of it you added some drops of growth factors. This pink color flower kind of thing, those are not flowers, those are growth factor molecules. And here, this interaction, it is showing the interaction between the growth factor and the scaffold, which is allowing absorption to happen. Okay, so it can be due to like hydrophilic interactions, which can be supported. Okay, charge based interaction, ionic interaction, etc. And the third way is the layer by layer. So here's, yes, a green color is a positively charged. Okay, and this is also like different charges is given, so, different color. So green can positively charged, blue can be negatively charged. The order of polyelectrolyte can be changed, and as they are varied, you are sandwiching, like you're making a sandwich, or you are basically trapping your growth factors in between. So these are the three physical encapsulation techniques. Then another way of basically encapsulating your or immobilizing your growth factor onto scaffold is through chemical conjugation. So previous one we saw a few examples of physical conjugation. Now here chemical conjugation. So we can take help. We can use various reactions. Okay, like coupling reactions. The first is the carbodiamide coupling immobilization. So carbodiamide is a functional group basically, and this functional group has been used to covalently cross-link or covalently bind the growth factor molecule onto the scaffold. So cross by the term cross-linking, what we mean? We mean some links has been, some linker chemical molecules has been created. Okay, so and this molecule is here in this case is a carboamide group. All right, the advantage is it is simple. So the reaction that is going to promote this carbamide based coupling, it is easy, and it helps in very high conjugation ratio. Means this reaction is successful in conjugating large amount of growth factors onto the scaffold, and the cost of doing this reaction is low. So it is affordable by most of the people. Now the disadvantage is during this coupling, you may like attack the other chemical reaction that is going to happen. It may attack the active sites on the growth factors. So you may alter the structure of the growth factor and thereby its functionality. Okay, this is the disadvantage. Then another way of chemically conjugating or basically immobilizing your growth factors onto your scaffold using chemicals like through covalent bonds is through using a chemical called polydopamine. So this chemical is very very similar to nature to a chemical which is naturally found in nature is dopa. This dopa chemical is synthesized by the organisms, mussels, okay, and they're found on the beach. So through these dopa chemicals, mussels can very strongly adhere on the surface. So people studied this chemical and developed a chemical synthetic version of it that is called polydopamine, which is having the same properties as the dopa released by the mussels. So polydopamine, if you coat any surface, any scaffold surface with polydopamine, then that surface becomes very adhesive in nature. Means anything can go and adhere to it or bind to it very strongly. So the growth factors can also like strongly bind to a surface which is coated with a polydopamine layer. So in this way, we can do chemical conjugation. So there are various advantages. First is the addition between the growth factor and the polydopamine coated scaffold is quite strong. Okay, and and you can actually coat any type of surface. You can coat any hydrogel surface or a metal surface with polydopamine, and then allow your growth factors to adhere on it. So to all types of surface you can use the binding that is happen between the growth factor and the scaffold is reversible. So it is not like at the site your growth factors won't be released. Okay. So here strong adhesive bonds are there. Okay. So that is why reversible is possible. Any chemical covalent bonds are not being formed. Okay. Please note this. That's why it is reversible. Only thing is, this unique chemical can, even though it is not chemically binding, but this unique chemical offers stable binding and and provides affinity to the growth factors to strongly bind to the surface. Okay. Disadvantages, growth factors can lose its functionality. Means depending on from which site or from which angle. So growth factor is a 3D molecule. So if the binding is happening in such a way that its active site is masked, okay. So again the functionality or the site through which is going to bind to the receptor, if that is masked by the strong adhesion onto the polydopamine coated surface, then it won't be able to signal the cells, right, and won't be able to give you the desired effect. So this is this diagrammatic representation. So here for the carbamide coupling reaction, people use these two chemicals, EDC and NHS. The full form is quite big. Maybe you can Google it. So this particular in presence of this particular combination of chemical, carbamide coupling reaction is possible. Okay, and if you coat a surface with polydopamine, all right. So it is basically you can see OH groups are there. So hydrogen bonds can happen with the growth factor molecule containing, say, ammonia group with hydrogen atoms on it. So you can basically link your growth factors on the polydopamine coated surface through hydrogen bonds, and these hydrogen bonds which are formed are very stable. Okay. Now another way of like immobilizing your growth factor onto a scaffold is taking inspiration from how the growth factors are present in the ECM. So in the ECM, we have a large number of molecules, and many of the molecules, you know, are like collagen, fibrin, etc., which actually compose the ECM. But there are also the small molecules, which are growth factors, which are often linked with these ECM molecules. So it acts basically as a reservoir of growth factors. and we can basically uh, like modulate your our ECM, like how the ECM is going to affect your cellular behavior if we change the growth factor composition on it, right? Then so taking this knowledge in hand, like how actually growth factors in the say nearby environment is adhering on the ECM, taking this, like how this mechanism is happening and mimicking this in vitro, we can immobilize our growth factors. So first approach in this method is using heparin sulfate. So heparin is a ECM molecule, and it has been shown that heparin can strongly bind with growth factors like BMP2 and VEGF. There are other growth factors as well. So if we coat our surface with this heparin sulfate molecule, then we can ensure that this particular classes of growth factors will only strongly bind on our surface. And if our purpose is to like deliver these growth factors, then we will be successful at it. So advantages, we are mimicking the ECM architecture in this way, and we are also going to get better control over how because the interaction between the growth factors and the heparin molecule we know, all right, like we have seen in nature, like in the tissue environment. So we can actually then understand how the growth factors are released, okay, and we can basically control. From this understanding, we can basically control the release both spatially and temporally, all right. Then adhesive based adaptation is okay. I think I disconnected for a second. So I hope I didn't miss anything. Am I audible?
Yes, ma'am, audible.
Yeah. So yeah. So another approach is like in this class is using proteins, ECM proteins like fibronectin, fibrinogen, collagen instead of heparin sulfate. So we can take help from the literature to understand which type of growth factors strongly bind to these ECM proteins, and then make a scaffold either using collagen, fibronectin, fibrinogen, etc., or we can coat our scaffold with these proteins and um, only allow certain type of growth factors to adhere on a scaffold. So people basically have reported, for example, BMP2 is MP. So BMP2, it has a very high affinity to bind on fibronectin fragments. So we can, rather than using fibronectin, is a very big molecule. So rather than using the whole fibronectin molecules on the scaffold, we can use its fragment, all right, and immobilize those fragments on the scaffold, and then allow only BMP2 to adhere. So this is another way inspired by ECM to encapsulate or immobilize your growth factor. And then the third example of doing this is we can mimic the hierarchical structure of the ECM, okay, in our scaffold. So how the ECM architecture is there in the tissue, it plays a very important role in modulating the growth factor activity. So, in one study, what they did, they created chitosan and collagen scaffold, right? And the scaffold was created in such a way, like the arrangement of the ECM and the collagen was like mimicking the ECM structure of bone, all right? And when they did that, they were able to basically control where the BMP2 growth factor was arranged. So they took inspiration from the ECM of bone, right? So bone contains a large amount of collagen. So they took inspiration from that and saw the arrangement of different molecules, like the collagen and other growth factors, then tried to mimic it in vitro condition using the using chitosan and collagen, and trapped the BMP molecules the way it is there in the bone. So when they use this artificial tissue and for bone regeneration purposes, they saw very good results. Like regeneration was happening. So bioactivity and release kinetics in this way, the of the immobilized growth factors is can be very well controlled. Okay, and it basically depends on how the growth factors are adhered, or how strongly like how much affinity it has towards the structure, micro nano level structure that we have created. Okay, so in these figures, again, the three ways through which people are taking inspiration from ECM to immobilize growth factors is shown. So we can go coat our growth factors with heparin. So this blue molecules is heparin, heparin sulfate, or we can use ECM proteins like collagen, etc., to coat our surface and onto which the growth factors can bind, or we can create a architecture, micron level and nano level architecture using various ECM components. Okay, and then like allow the growth factors to distribute on this structure. Okay, so in this way, uh, we have looked into how immobilization of growth factors onto the scaffold can be achieved. Now, the next way of delivering growth factors is using nanoparticles. So, I hope you understand what are nanoparticles. These are basically particles which are having sizes in the nanometer ranges. All right. So, people have used a lot, like these nanoparticles have been used a lot in the area of drug delivery, because these are very small, right, in the nanometer ranges. They, because of the small size, they have a very high surface area to volume ratio. Okay, surface area to volume ratio, and thus high surface area to volume ratio will allow high amount of drug loading. Means you can the payload that you can deliver using nanoparticles is quite high, and depending on what type of material you are using to make these nanoparticles, you can make it a smart nanoparticle. Means it can respond to a particular type of environmental stimuli, like change in temperature, pH, or magnetic field if it is there, then ultrasound, etc. These are various kind of stimuli which can modulate the nanoparticle behavior. Now we can also control how the nanoparticles are distributed spatially. So which is this spatial, this ability to control spatial distribution is big, which is why they are used for delivering drugs. So like growth factors, drugs, if they are delivered at a very high concentration, it can be toxic, right? So we want spatial like control over it, delivery, special like control over spatial distribution. We want that. So if we, because of the same reasons, we will also use the nanoparticles as a carrier to deliver the growth factors. Then again, due to encapsulation within environment, closed environment, the growth factors will be protected from the body's physiological environment, and thus degradation will be prevented. Okay. So these are various types of nanoparticles which have been used for delivering growth factors and is extensively researched in, and you can find them in literature. These are the various types. But in our today's lecture, we will discuss only few of them. Okay. So first type is the synthetic polymer nanoparticles. So here you are making nanoparticles using synthetic polymers like PLA, PLGA, PGA, and there are others as well. Okay, so using a synthetic polymer, not a natural polymer. You know the classes of polymers that we have discussed previously. So, and these polymers have been widely used to make nanoparticles for drug delivery application. They are considered safe through extensive research, and also the polymer PLGA is FDA approved. Okay, so you can use this polymer and show its application in vivo as well and go for clinical trials. And it protects the drugs inside, obviously, because it is protecting the growth factor, the drug from the degradation, physiological environment, which can, it, in presence of physical environment, it can happen. But one problem with using a polymer like PLGA is that this polymer has very low binding affinity to proteins at physiological conditions, that is 37 degrees Celsius and pH 7.4, because of which whatever protein that you load within the PLGA nanoparticles, it will has a very high chance that it will start to leach out. Okay, so if you are so you won't be able to deliver your intended concentration of growth factor at the target site. Okay, now you can use various type of proteins, like natural proteins which are available in our body, and use and make nanoparticles out of those proteins. So a few examples that have been used for this application is the use of albumin, and then BSA, bovine serum albumin. So these proteins have been used to make nanoparticles. Okay, there are various advantages to it. First, these proteins they themselves has binding sites, and you can use them to conjugate your drug. You conjugate your growth factors. Then, if you can further coat these like protein nanoparticles with charged polymers like polyelectrolytes. So if you do that, it means if you want to do that, it is possible. Okay. Then this material like albumin, these are biodegradable, so often they are non-toxic, biocompatible, and you can it is also easy to fabricate albumin-based nanoparticles. But one problem with protein-based nanoparticle is they have a very high tendency to aggregate. Okay, they have a very high tendency to aggregate. So why aggregation can happen? Can anyone like think over it? We have actually discussed in the biomaterial part about hydrophobic surfaces, hydrophilic surfaces, and then protein adsorption, right? So maybe from that you can think over like why um, protein-based nanoparticles can aggregate. If maybe not now, maybe you can answer in later class. All right. But to prevent this aggregation, if a colloidal solution is not formed, rather the nanoparticles aggregated, then what will happen? It will not freely circulate in the blood, right? And it through the flow of the blood, it will not reach your target site. Okay, so we don't want aggregation. So which is why we stabilize it. We stabilize the nanoparticles by coating it with something else like glutaraldehyde or polysaccharide. So in this slide, I have given an example from a study where they have used BSA, bovine serum albumin nanoparticles, to deliver two different types of molecules, basically BMP2, which is a growth factor, and I think vancomycin is a drug. Okay, so they have delivered it together. Okay, so what they have done, they have first conjugated the BMP2 with BSA. So through the active sites on the binding sites on the BSA, the BMP2 could bind, and then these BMP2 loaded nanoparticles, it was given a coat of a polysaccharide, that is chitosan, right? So chitosan coating gives the overall charge of the nanoparticle to be positive. Then they made another type of nanoparticle. Now in this case, they encapsulated the vancomycin, all right, and made vancomycin loaded nanoparticles. Now here, this nanoparticle was given opposite charge. How did they do that? They first coated it with chitosan so that it becomes positively charged, and on top of it, they coated it with oxidized alginate. So oxidized alginate gives it a overall negative charge because of its carboxyl groups. Now these two types of oppositely charged nanoparticles, they were layered, all right, on top of a titanium surface. So this titanium surface was first coated with polydopamine, all right, and made like highly adhesive in nature, and then on top of it the oppositely charged nanoparticles are like assembled in a layer by layer manner. So when they tested how the BMP2 and vancomycin was releasing from this structure, they saw that it is happening in a sustained manner and over a period of 30 days. So basically what we saw no in the topic where we were discussing that people are mimicking the ECM architecture, right? And encapsulating the growth factors according to how it is there in the native ECM. So that logic or that idea is basically combined here, but with the idea of encapsulating growth factors within the nanoparticles. So it's layer by layer assemblies often seen in ECM. So they're basically combining the two approaches to get a sustained release of their growth factors. Any queries?
No, ma'am.
All right. Now we can also use, rather than using the proteins, we can also use polysaccharides. So polysaccharides as a material, they are abundantly found in nature. They are of very low toxicity, highly stable in structure, then cost is low, so we can use it as much as possible. Then biocompatible, it is means often useful or can be used for different applications and will give you the desired result. And the polysaccharides will have various type of functional groups on it, the chain of the polysaccharide, which you can use to conjugate your any molecule of interest like growth factors. So in this example, they have made chitosan is a polysaccharide, so they have made chitosan conjugating sulfate nanoparticles to encapsulate the growth factor BMP2. So what they have done, they have seen like heparin sulfate, chondroitin sulfate can also bind with BMP2 because it has a high affinity to do so. Okay, so we discussed previously, no, heparin sulfate can do it. So similarly, BMP2 can also do it. So first, then they prepared a chondroitin sulfate and BMP2 conjugate. Then this conjugate was encapsulated over using chitosan. So chitosan is positively charged, okay, and chondroitin sulfase conjugate is negatively charged. So in this way, due to electrostatic interaction, the encapsulation by chitosan is possible, and they made nanoparticles with chitosan coating, and in core there was BMP2 and chondroitin sulfate conjugate. Okay, so in this way they prepared. This is an example of polysaccharides based nanoparticle. Now you can use other type of nanoparticles as well. So liposomes are there, mesoporous silica we can also use. Then in this particular figure, you can find other types. You can use polymer nanocapsules. Okay, the lipid based one is the liposome one. Okay, so we have discussed synthetic polymer nanoparticles, we have discussed protein based nanoparticles and polysaccharide based nanoparticles. We have not discussed the nanocapsules. Okay, polymer nanocapsules we have not discussed, and the center one here, this is also not discussed, where the in nanoparticle and loaded growth factor loaded nanoparticles, they are somehow linked, chemically conjugated or physically encapsulated within the scaffold. So we are not encapsulating the growth factor, we are rather encapsulating growth factor loaded nanoparticles within the scaffold. So these two, G and F, we are not discussing. We are briefly touch upon the liposomes part and mesoporous silica nanoparticles. So liposomes, the structure of the liposome basically is very much similar to the cellular membrane, where there is a phospholipid bilayer. So liposomes are made in similar way, where phospholipid structure, the shell structure is made out of phospholipid bilayer, where there is one end is hydrophobic and another end is hydrophilic. So these type of structures you can use to load either hydrophobic or hydrophilic drugs or proteins, okay, within the core of it. The use of this liposome is preferred because people can people have seen that they are biocompatible for various applications. Then they are not very toxic or non-toxic, but only problem is again that they can aggregate under aqueous conditions. So the hydrophilic part, maybe the hydrophilic part of the lipid bilayer, it can strongly interact with the aqueous environment and thus can cause aggregation. So this problem can be overcome by giving a coating like of a synthetic polymer like PLGA or PEG, etc. Or you can give a coating of polysaccharide, okay, and prevent the aggregation problem. Then mesoporous silica, okay. So mesoporous means the pore size which is greater than 50 nanometers, means very big pores in the micron range, all right. So and silica nanoparticles with mesoporous porous structure. So these nanoparticles are themselves like highly porous. Materials can easily come in and go out. Okay, it is not properly, whatever loading that you're giving inside, it is not properly enclosed. The load can always come out and released into the environment. So as but there are advantage of these nanoparticles, which is why people are using it. You can control the particle size very easily. So silica nanoparticles, it is very easy to control the particle size. You can create nanoparticles of various sizes, and they are very biocompatible for various applications. So as I said, the porous structure is there, no? So there is a very high chance that the loaded growth factor will instantaneously release, means there will be a burst release. So you can eliminate that by giving a polymer or polysaccharide coating over it. Okay. Now the last approach that we're going to discuss on how to deliver growth factor is the gene-based approach. So here what we are doing or what we are thinking of doing is rather than delivering the protein, we will deliver the gene encoding that particular growth factor. So we can either deliver RNAs like siRNAs, or we can deliver DNA, mRNA, so any nucleic acid. Okay, so the intention of that nucleic acid, it can either go and suppress the gene expression of particular growth factor, or it can promote its gene expression. Okay, so within the cell, we are modulating the growth factor concentration depending on what kind of behavior we want to see. So this method that we are discussing here, it is relatively more complex than the other methods that we have seen. All right. So how we will do gene delivery then? First thing we have to do, if we are using a nucleic acid which is very long base pair, so we have to first condense it, make it smaller so that it can fit into a nanoparticle. Then we have to prepare nanoparticle and then load the condensed nucleic acid within the nanoparticle, but we have to make sure that there is no nucleases. Otherwise, your nucleic acid will degrade. Then the nanoparticle which is loaded with your nucleic acid, it has to be allowed to allowed the cells has to be made more competent so that it can take up those nanoparticles loaded with gene. All right. And usually the mechanism that people want to go through is endocytosis. There are other mechanisms as well, so you can maybe search it. So through endocytosis you can trigger the uptake, means entry of these gene loaded nanoparticles inside the cell. So endocytosis, when it happens, endosome is created. Okay, naturally endosome is created in the which floats in the cytoplasm. So what will happen within your endosome, your nanoparticle will be present. So the next step would be then to escape this endosome. Okay, and the nanoparticle will now will freely circulate within the cytoplasm. Now, if you want to if you want a nucleic acid to release in the cytoplasm, all right, then it's fine. It is easy. All right, then your job is done. Nucleic your nanoparticle is there in cytoplasm. Then you have to engineer nanoparticle in such a way that the payload is released. But if you want your gene to or your nucleic acid to go and release only at the nucleus so that it can bind with the host DNA, okay, and integrate with it and get expressed along with the host genes, then you have to use some alternate approach. So the nucleic acids which are released or is generally released in cytoplasm are the siRNA. So I hope you're familiar with RNA interference mechanism, where siRNA is used to block protein synthesis, like to block translation, because siRNA can bind with mRNA and make it double stranded, thus prevent the translation mechanism from reading that particular mRNA. So if you're using siRNA, then you will want the nanoparticle to release those siRNA in the cytoplasm itself. Otherwise, if you're using DNA, okay, which is encoding directly the protein, say, then you have to go into the nucleus. And the last step would be your cargo is released, right? Means the cargo means the nucleic acid is released. But what about nanoparticle? Okay, it cannot just stay there in cytoplasm. So you want to use the material to to biodegrade. Okay, and when it undergoes degradation, it should not cause toxicity to your cells. So these are few steps on how to deliver gene. All right, and through that gene delivery, we are going to basically control the expression gene growth factor expression. Okay, and thereby moderate cell behavior. So various vectors are basically used to deliver your gene. You can use either the viral vectors, but the disadvantage of using viral vectors is that they safety is a concern. There's always a chance that some mutation in the virus happens and it becomes virulent. Although we have previously removed the virulent component, okay, and the amount of gene that you can deliver using viral vectors is less, but the efficiency is very high. Means you will be able to deliver your gene at the target site highly efficiently. Means every time your or most of the time your gene will be delivered at the target site, intended site. Now if you use non-viral vectors, they are less immunogenic, so you don't have to worry about the virulent factor, okay, or triggering the immune system. It won't trigger the immune system that much. But and also another advantage is that payload is high. So often the small amount of nucleic acid, if you are worried that it won't cause the intended change, so you can load higher amount. And if you're using non-viral vectors, then it is possible, but efficiency is lower. Means targeted delivery at the site is not possible. Like it is not happening every time. Okay. Now, uh, it is difficult to deliver genes using either of the two types of vectors in vivo. Okay, so generally, what people are doing now, they are taking the cells out from the patient, okay, from the in vivo environment and culturing them ex vivo, that means outside, and then these cells are transfected with the viral vector non-viral containing your gene of interest, and these cells after getting transfected with the gene of interest, they are put back, okay, into the in vivo environment. So in this way, gene delivery or gene therapy, etc., is being done. So that's it for today's class, and these are some of the references which I used for today's class. Maybe you can go through them later on. And if you have any queries regarding any topic that we discussed today or maybe previously, you guys can let me know. Any queries?
Okay, ma'am.
All right.
No, ma'am.
Okay. So